Good afternoon, everyone, and welcome to this session on COVID drug discovery. My name is Professor Monica Slavin, and I am head of the Department of Infectious Diseases here at Peter MacCallum Cancer Centre. First of all, I would like to acknowledge the traditional custodians of the land on which myself and today's presenters and audience are located. I would like to pay my respects to the elders, both past, present, and emerging, and extend that respect to other Indigenous Australians in the audience and online. So before we kick off today's program, there's a couple of housekeeping notes. Please note today's webinar is being recorded and will be made available to view on the VCCC Alliance Open Learning page once the final data is published. If you have any questions for today's presenters, you can do this via the Zoom Q&A function. Please submit your questions throughout the presentation and we'll aim to answer all the questions at the end of the session. So today we have a fantastic and very exciting program about discovering antiviral treatments. First speaker will be Professor Sharon Lewin. And Professor Lewin is the inaugural director of the Peter Doherty Institute for Infection and Immunity, a joint venture between the University of Melbourne and the Royal Melbourne Hospital. She's an infectious diseases physician and a basic scientist. Her research focuses on understanding why HIV persists on treatment and developing clinical trials that ultimately aimed at finding a cure. She's published over 300 publications and given over 100 major invited talks on HIV. She's president-elect of the International AIDS Society and co-chairs the Global Advisory Board on Towards a Cure for HIV initiative. She also leads the Australian Partnership for Preparedness for Infectious Diseases Emergencies, a prize, an NHMRC-funded Centre for Research Excellence. Scientists from the Doherty Institute were the first to isolate and share SARS-CoV outside of China in January 2020. And the Institute continues to work on all aspects of diagnosis, therapeutics, and vaccines for COVID-19. The other two speakers I'll introduce now because we will move straight on to their talks. So second will be Dr. Wei Zhao, who's a postdoc research fellow specialised in HIV and hepatitis B co-infection in Professor Sharon Lewin's laboratory at the Doherty Institute, with experience in genomics, cellular biology, biochemistry, and more recently, SARS-CoV-2 virology. His main research interest is in vitro study of HIV and Hep B co-infection. He has a 5-year experience working in high containment laboratory, and recently he established a new collaboration with Dr. Mohamed Fareh to develop novel gene scissors for potential treatment of COVID-19. And that leads me to introducing Dr. Mohamed Fareh. So Mohamed's a senior research fellow with the multidisciplinary background in RNA biology, CRISPR, cancer, and advanced single molecule biophysics. He obtained his PhD in 2012 in France, where he uncovered a subset of non-coding RNAs that regulate cell plasticity in high-grade brain tumours. He did his postdoc training in the single molecule biophysics lab in the Netherlands, where he pioneered new single molecule approaches to investigate the biology of non-coding RNAs and CRISPR effectors. In August 2018, Mohamed relocated to Australia to join the Trapani lab here at Peter MacCallum Cancer Centre in Melbourne. He leads projects focusing on reprogramming RNA targeting CRISPR tools called CRISPR-Cas13 for transcriptome remodeling. His team is interested in understanding the molecular basis of RNA recognition by Cas13, reprogramming Cas13 to silence undruggable tumour drivers and viral transcripts. So now it's a great pleasure for me to hand over to Professor Sharon Lewin to start this exciting program today.
Great, thank you very much, Monica, for the lovely introduction, and a great pleasure to be here. And present on behalf of this team. So we've been doing some very interesting work over the last 20 months or so, as you've heard about, in great collaboration between the Doherty and the Peter MacCallum Cancer Centre, primarily driven by Mohamed Fareh's initial idea, together with Joe Trapani, and work that's involved both myself and Wei. So I'm going to try and set the scene a bit for you to tell you a little bit about treatments for COVID-19, why we need antiviral drugs, and then we'll hand over to Mohamed and Wei that will tell you the story of the role CRISPR-Cas13 might play. So, as you all know, the pace of science has just been incredible for COVID-19, and we've all reaped the benefits of that with vaccination. This comes from my favourite coronavirus journal, the New York Times, and the current updated total number of people that have received a COVID vaccine worldwide, 6 billion on September the 24th, 2021. However, although vaccines and the pace of discovery has been unprecedented, the advances in treatment have been much slower. And so today I'm going to talk to you a little bit about why, and to understand why, I'll tell you a bit about the natural history of COVID-19 and the opportunities for treatment. I'll tell you what we've got, where we've landed so far with monoclonal antibodies, a little bit about direct-acting antivirals, and then hand over to Mohamed and Wei to tell you about CRISPR-Cas13 and gene editing as a novel approach towards developing an antiviral drug. So, natural history of COVID-19, I suspect you all know this now, but COVID-19 is a complicated illness. It's not just a single illness. Early in infection, most of the pathogenesis is largely driven by high levels of SARS-CoV-2 replication, primarily in the upper airways or the nasal passage and in the lower pulmonary tract. And over time, usually within the second or third week, if there's disease progression, there's a change in pathogenesis away from viral replication and towards an aberrant and accelerated host inflammatory response. So because the disease is so different, you can imagine that the interventions that you might use early in infection would be very different to what you'd use late in infection. And this took a while to work out. So early infection, people have mild disease, they may have a dry cough, upper respiratory tract infections and fever. If they progress, they'll then move on to developing shortness of breath or some systemic evidence of infection such as transaminitis. And then if disease progression is significant, and only of course a subset of people will progress to this stage, they'll develop respiratory failure, shock, and require invasive ventilation. So you can imagine that the treatment goals at each stage of disease are very different. Early in infection, ideally you could treat to reduce the chance of disease progression and also potentially reduce transmission, because if you lowered the viral load in the nose, you'd reduce the likelihood of transmission to another person. In that intermediate or pulmonary phase, you want to reduce disease progression and potentially hospitalisation. And finally, of course, at this late stage, you want to reduce morbidity and mortality. So over time, as in the last 20 months, we've realised that the treatments that you implement at this phase are largely to dampen down the immunopathogenesis and reduce hypercoagulation, a feature of COVID-19. And early in infection, you want to enhance immunity or clearance of the virus and primarily use antiviral drugs. The real challenge is getting in early enough with a cheap and easy to use antiviral drug, where there's been very limited development, unfortunately, over the last 20 months. So there's been many therapeutics investigated, but few approved as treatment of COVID-19. These include antivirals. You've heard a lot about these ones that basically went nowhere: hydroxychloroquine and lopinavir, ritonavir. Ivermectin still under investigation, but absolutely no evidence that it currently plays any role clinically. Nitazoxanide is another potential antiviral, and really the only licensed antiviral is remdesivir, that I'll talk a little bit more about. Anti-SARS-CoV-2 monoclonal antibodies have also played a really interesting role that I'll talk more about, primarily working as an antiviral drug. And then we've had some progress with immunomodulators, really quite significantly reducing the risk of mortality should you end up with severe COVID-19 and be hospitalised. However, few COVID therapies are currently approved, and it's also approved in Australia. And most of the treatments that are approved are for moderate, severe, or critical illness. So these are the drugs that we use currently for the treatment of COVID-19: steroids, highly effective in reducing mortality; anti-IL-6, remdesivir, the only direct-acting antiviral drug; and another anti-inflammatory drug, a JAK-STAT inhibitor, baricitinib. And these therapeutics have been extensively investigated in many large phase 3 studies, and they have a role to play largely in people with moderate to severe illness and requiring oxygen. And what we use in people who are ultimately on mechanical ventilation differs a little bit. Now, antibodies have been really interesting to follow. When they were first introduced, they were tested in people with severe illness and they had no role. But as we've got better at understanding the pathogenesis of COVID-19, it's now apparent that monoclonal antibodies really do have a role to play in mild infection, and therefore they're now licensed for the treatment of mild infection here in Australia. And just some very recent data demonstrating that they may have some role to play in moderate infection in people that don't generate their own antibodies. However, the real dearth of interventions is in people with mild disease to stop disease progression. And ideally, we need a direct-acting antiviral agent that could be used to stop disease progression and also potentially as pre or post-exposure prophylaxis. So I've got a lot of experience in this area from the HIV world. We have fantastic antivirals for treatment. You can treat at any stage of disease and they work brilliantly, very different to COVID-19. And these same antiviral agents also work as pre-exposure prophylaxis to prevent you getting HIV, and post-exposure prophylaxis if you've been exposed to HIV. So if we had something like that for COVID-19, this could be a real game changer. So what do we know so far about monoclonal antibodies? Well, first of all, monoclonal antibodies are actually identified from people who recover from COVID-19, where we can isolate envelope-specific or spike-specific memory B cells. They then can undergo single-cell RNA sequencing, which identify the actual antibody sequence, which allows you to generate large amounts of antibodies that can then be used as a therapeutic. You can test these antibodies and their potency of inhibition against a range of COVID strains. So the world was very quick to develop antibodies. A lot of this technology was optimized in HIV. We have a lot of HIV antibodies, none yet licensed or used for treatment or prevention. But in COVID-19, exactly the same techniques were used, and we have a lot of antibodies that are actually quite potent and now licensed and being used. So this just shows you one publication of 2 antibodies that got all the way through to commercialization, one from Lilly and another from Regeneron. And it shows you the IC50, or capacity of the antibody to neutralize a live virus based on against its antibody concentration. So an EC50, the concentration at which you get 50% neutralization, the lower the number here, the more potent the antibody. So you can see here that these 3 antibodies worked pretty well against the original, what we call the ancestral strain, or the Wuhan strain that we had initially circulating in Australia last year. The next variant of concern to appear was the Alpha strain that came from the UK. And you can see these antibodies work pretty well against both Wuhan and Alpha. But then the Beta strain came along. This was the South African strain, and you can see that these antibodies no longer work against the South African strain. So very potent against some strains, not so good against other strains. So not easy to use clinically because you need to know what strain you're dealing with. And I mean, right now we know we've largely got Delta, but still not an ideal antiviral drug. However, these drugs, antibodies, have all gone on to become licensed. The first 2 were, they've got these impossible names, bamlanivimab and etesevimab from Lilly. They were initially designed against the Wuhan strain. They were rapidly licensed by the FDA, now no longer used because they're not active against subsequent variants of concern. The next 2 antibodies were made by Regeneron. They're used together, and so therefore they have a broader range of activity. They also have emergency use authorization, often referred to as Regeneron combo. Sotrovimab is the only antibody licensed in Australia. It's actually interesting antibody. It's active. It binds a different part of the spike protein to the Regeneron or to the Lilly antibodies, and it's active against all variants, at least in vitro, including Delta. Recently approved by the TGA to be used in Australia, and really very effective in people with mild disease. The antibodies have been also evaluated for prevention and treatment of mild, moderate and severe disease, and they've largely shown to have activity in mild and moderate disease, but have no role in severe disease. So antibodies are certainly becoming part of our toolbox for managing COVID. We're using them currently in Australia for treatment of mild disease in people that have symptoms for less than 5 days and have a high risk of disease progression, meaning they're over 55 or have comorbidities. But they're expensive. They're about $2,000 an infusion. They're a hassle because it has to be intravenous. And although they do work, they're not easy to implement. So this is the data supporting the use of sotrovimab in high-risk patients. This was a phase 2/3 study done by GSK. It enrolled people over the age of 18 who had COVID-19 diagnosed. They had less than 5 days of symptoms, so they're definitely in that early virological phase. High risk of disease progression defined as greater than 55 years of age and at least one other comorbidity. And you can see here when they compared sotrovimab to placebo, there was an 85% reduction in the likelihood of going to hospital or dying. So very significant impact if you can get sotrovimab into people within 5 days of symptoms. Combination antibodies can also prevent infection. So this is a really interesting study of the Regeneron combo, where participants over the age of 12 were enrolled within 96 hours after a household contact received a diagnosis of SARS-CoV-2 infection, and they received 1,200 milligrams of these 2 antibodies now, or matching placebo, and actually was given subcutaneously as 4 separate injections. That's a major advance, much, much better than giving intravenously. And again, worked really well. So this is this study was done prior to vaccination, but it's very actually relevant to people, particularly immunosuppressed people who have had an inadequate response to a vaccine, whereby administering the antibodies within 96 hours to a contact dramatically reduces your chance of acquiring COVID by about 80%. So quite promising results. Antibodies work. They work as antivirals. They work if you can give them early enough in infection or after exposure, but they're a bit of a hassle and they're expensive. And ideally, for a perfect outpatient therapy, we need something that will achieve favourable clinical outcomes, so prevent disease like the antibodies do, reduce viral load and onward transmission. We don't yet have the answer to that, whether antibodies do that. Obviously, reduce symptoms, reduce hospitalisation, and maybe prevent complications like post-acute SARS-CoV-2 infection or what you would know as long COVID. You want a treatment that retains efficacy against all strains. So that's why antibodies aren't perfect, because these strains continue to evolve in that exact area where antibodies are targeted. And ideally, these have to be safe, well tolerated, easily administered, ideally oral, and available and distributed and at minimal cost. So antibodies don't do well on this large last area. And that's why there's now this very high interest in developing direct-acting antiviral drugs. And in fact, there's such a high interest in this that the National Institutes of Health recently announced an antiviral program for pandemic RNA viruses with a $3 billion investment in order to develop drugs that directly target RNA viruses of pandemic potential, because together with vaccines, this could really be quite revolutionary, but you need the intervention to be cheap, oral and accessible. So there are many, many targets in the SARS-CoV-2 life cycle that are amenable for drug discovery or antiviral development. And this just shows you the life cycle of SARS-CoV-2. The details aren't important. The virus enters, undergoes a series of steps, which involves a number of enzymes, particularly protease and something called RNA-dependent RNA polymerase to make new RNA. The particles then get packaged, undergo maturation and release from the cell surface. Now in HIV or hepatitis C or hepatitis B, all viruses where we've got good antivirals, we target lots of these steps by actually targeting the viral enzymes to stop doing their work. So there are many targets for direct-acting antivirals for SARS-CoV-2, blocking entry, blocking protease, blocking replication or polymerase maturation and release. And a lot of work happening in this area. But we only actually have one approved antiviral for COVID-19. It's a repurposed antiviral, remdesivir. It blocks RNA-dependent RNA polymerase. So this step here, it was originally developed for Ebola, has got modest activity against COVID, but it's a hassle because it can only be given intravenously and hasn't really shown dramatic benefit for hospitalized patients, although we still use it. So we've learned in the pandemic that developing small molecules and identifying the target, often needing structural biology, then using rational drug design, then testing the small molecule, assessing toxicity, and manufacturing a brand new molecule takes time. And in a pandemic, there isn't the time. And this is why we're in this position now, 20 months later, with a ton of vaccines available, but no direct acting antivirals. There is, however, one antiviral that is looking quite promising. That's Monopyravir. It's a polymerase inhibitor. And in a recent placebo-controlled study, the results are here, where participants were enrolled within 7 days of symptoms and treated for 5 days with different doses of Molnupiravir, placebo, 200 milligrams twice a day, 400 milligrams twice a day, or 800 milligrams twice a day. You can see that clearance of virus tied to clearance of SARS-CoV-2 was accelerated in participants who received 800 milligrams BD. And you also can see that at the 5th day, the number of people that had infectious virus in the nose was greatly reduced compared to placebo. And interestingly, there's only been a press release of a phase 3 trial in India of 800 milligrams BD compared to placebo, but this intervention reduces hospitalisation. So this could be the first simple oral antiviral drug. Many others in development, but nothing yet close to Molnupiravir. However, you could have a totally different approach to direct-acting antivirals, and that is to, instead of targeting a structural protein or an enzyme, you could directly target the RNA. Either genomic RNA, what we call, which is the sequence of the actual virus, or subgenomic RNA, parts of the virus that make the relevant viral proteins. And this, of course, now can be done. And it can be done with something called CRISPR-Cas13, which you're going to hear a lot more about. And we've been evaluating CRISPR-Cas13 as an antiviral. The work started back in March 2020. It was Mohamed Fareh's idea. Mohamed and Joe came to me back in March 2020 and said, what do you think about using CRISPR-Cas13 as an antiviral for SARS-CoV-2? And I said, what's CRISPR-Cas13? I'd heard of CRISPR-Cas9, but not of CRISPR-Cas13. But now I've become quite obsessed with it. And I really do think this is an incredible platform for novel antiviral drug development. So we've been working together with Mohamed and Joe and Wei, who rapidly turned from his work on HIV and hepatitis to focusing on SARS-CoV-2, and they're going to tell you their story about CRISPR-Cas13 and how it may fit in to become an easily administered antiviral drug. So I'll stop there and I will hand over to Mohamed.
Thank you, Sharon, for the lovely talk and introduction about the biology of SARS-CoV-2 and the therapeutics. I really learned a lot from your introduction. So I suppose this project started with really our fascination with the arms race between bacteria and bacteriophages. As you know, bacteria are these really tiny single cell organisms that they live everywhere in our environment, but they are basically prone to infection with viruses called bacteriophages, and bacteria are constantly challenged by these bacteriophages, and their survival is extremely dependent on their ability to fight these viruses and eliminate them. So throughout their evolution, bacteria developed something really fantastic called CRISPR, or CRISPR molecular scissors, which recognizes the genome of these bacteriophages or viruses, to simplify this term. And so it can recognize the genome of these invading viruses and cut it in sequence-specific manner, which is just extraordinary. We, like mammals, we lost this ability to do this throughout the evolution, and we evolved this cell-based immunity or adaptive immunity, while bacteria, they have this nucleic acid-based immunity. And you might have seen this in the news a year ago when CRISPR was awarded Nobel Prize of Chemistry to Jennifer Doudna and Emmanuel Charpentier for pioneering CRISPR-Cas9 as a genome editing tool that really started from this fascinating observation on the way bacteria fight their viruses. So likely the nature spoiled us with many CRISPR types and subtypes that they can target single-stranded DNA, they can target double-stranded DNA, or they can target RNA. And we are interested in a CRISPR type called Cas13, which, unlike Cas9, which is the rock star out there, targets RNA. It doesn't target DNA, it targets RNA. So what is really fascinating about this enzyme is that it's really a single protein. It's easy to purify, it's easy to engineer, and has really simple and one guide RNA, as you can see here in this schematic. So this guide RNA has this part called the spacer, and this is the most important part of my talk, is that to know how this spacer works. So the spacer within this guide RNA provide the specificity. So to enable Cas13 to target any RNA of interest, you need a match. You need the spacer sequence to base pair or to recognize the target. And that recognition is mediated by this RNA base pairing. It's really a match between like a key and the lock. And so what is really exciting that this spacer is very long, is 13 nucleotide long, and provides really high specificity. So the probability of finding anything with similar sequence in the host transcriptome-wide is zero because of this extensive length. And you can program this protein to target one gene or several genes. It's very cheap and easy to do. And so our work in the Trepani lab is basically to target gene fusion, to target tumor drivers in childhood and adult cancers, like gene fusions, overexpressed RNA, and point mutated RNA. And all these genes, basically, they drive cancer. But for the interest of time, I won't go into this part today, and I hope we'll have opportunity to tell you about our exciting work in the cancer space in the future. So I'll just go back to the COVID work and try to show you a little bit of data from this study. So our initial aim when this COVID pandemic started was, can we reprogram or repurpose this tool, this Cas13 tool we have, that we use it to target tumor RNA, to target SARS-CoV-2 strains? And can we develop a molecular blueprints of antiviral therapeutics that can be used against future pandemics? And so it all started with the release of the genome, the genomic sequence of this virus. So when this pandemic started, the Wuhan lab that sequenced this, the genome of this virus, quickly shared it with the community, which really enhanced the research output from different perspectives. So when that happened, we just downloaded this sequence very quickly and designed we used bioinformatic tools to design basically thousands and thousands of guide RNA that are all potentially good guide RNA to allow the suppression of this virus. So these are called single nucleotide tiled guide RNA that cover the whole genome of the virus from the 5' end to the 3' end, and there are 30,000 guide RNA roughly there. Then we used our selection criteria to select the most potent guide RNA that could allow us to achieve high silencing efficiency, all done bioinformatically. And then we went to the lab and tried to test whether those guide RNA work. So the first candidate we investigated is the spike, is a protein, viral protein that allows this virus to enter the host cells. And the fact that we are in Peter Mac and our labs are PC2 labs, so we don't have the confinement facilities to work with live viruses. So we developed this simple and clever system that allow us still to target part of viral genome. Here, in occurrence here is the spike. So we would fuse the spike with the fluorescent protein called the EGFP. And if we target the spike RNA, there will be degradation of EGFP RNA as well. And as readout, we just get loss of green fluorescence, as you see here. So this is just the workflow of this assay, how we do it. So we express the Cas13 and the guide RNA specific against the spike in the cells together with the spike transcript. So the guide RNA gets loaded into Cas13 and guide this target recognition in sequence-specific manner. Now, if we have degradation of target RNA, we have a massive loss of fluorescence intensity here. So I'm just showing you this data set, for example, with non-targeting guide, a guide RNA that doesn't match anything, doesn't have any specificity. We use as control. You see that you have a high expression of the spike protein here. But when we have a guide RNA targeting the spike, so here we tested 4 different guide RNA in 2 different cell lines. We have really high silencing efficiency that can reach up to 80 to 98% silencing efficiency. So we can now go to the lab and try to use this guide RNA as a drug, and we titrate the amount of guide RNA we provide to the cells. And you can appreciate here that we have really nice dose-dependent silencing that tells us that Cas13 and its guide RNA can act as antiviral and can silence the gene target in sequence-specific manner. So from the IC50 here, we could calculate the potency of this guide RNA, and you see this is highly potent with the affinity in the range of picomolar. So to demonstrate that this is really specific, an assay that we usually use is that we create a mismatch. So now we have the same guide RNA, but we introduce 3 nucleotide, 6 nucleotide, or 9 nucleotide mismatches at different position of the spacer sequence, and then we probe its specificity. And you can see here in this graph that the silencing efficiency of the wild type is really high, but you gradually lose the silencing efficiency when you introduce 3, 6, or 9 nucleotide mismatches at different positions. So this tells us 2 things, basically, that this is extremely specific, and the silencing of SARS-CoV-2 spike is really dependent on the base pairing between the guide RNA and the target. But it also tells us that few nucleotides, for example, here can be well tolerated. So this drug has really good balance between mismatch tolerance and specificity that we explored later on. I will touch on that a little bit. So because we could afford to design many guide RNA, as I said, they are really cheap, and we can make drugs against any virus in a week in the lab. Here we designed an experiment that allows us to probe 61 guide RNA that are tiled with single nucleotide resolution across this region of the spike. And so the key point here is that although most of guide RNA are very efficient, there are some guide RNA that are extremely potent, that shows really high silencing efficiency, but the guide RNA just next to it shows very poor silencing efficiency, which basically we are trying to understand how this enzyme works so we can reprogram it to silence any other virus in the future in an efficient way. Next, we move to silencing another gene of the virus, which is really important and well conserved. It's called the nucleoprotein, which is basically allow the packaging of viral genome and to package it inside this viral particles. And so we did the same design. Basically, we designed a 3-guide RNA here targeting the nucleocapsid. And you can appreciate here that we silence the RNA of the virus very efficiently. We silence also the protein of the virus, as shown here in this Western blot. So this data shows efficient silencing of nucleocapsid at the RNA and protein level, which was really promising to move it forward to silencing an actual virus. And you see that the affinity of this guide RNA we picked here to move it forward is at the range of sub-picomolar, which is extremely potent. So at that time, one concern that we knew about is the ability of viruses in general, but specifically RNA viruses, to evolve so they make mistakes when they replicate their genome. And these mistakes allow them to gain fitness and escape from therapeutics. For example, here we have an epitope on the virus that is recognized with monoclonal antibody. And if the virus accumulates the right mutation, it can escape this recognition by antibodies. And Sharon just nicely summarized how these monoclonal antibodies could escape, the SARS-CoV-2 could escape monoclonal antibodies recognition. So we thought, can our Cas13 basically be resilient to this mismatches? And at that time we were developing this study, there was really clear evidence that there are some strains that are just taking over the original Wuhan strains because they gain fitness or they gain escape from immunity. So to address this question, show this very quickly that we created the mismatches at different position between the spacer and the target. And those artificial mismatches basically mimics what would happen in real settings where virus mutate. So what if we create a mismatch between guide RNA and the target? Would this compromise the ability of Cas13 to recognize its target and silence it? So the short answer is no. So you see with all this guide RNA we designed here, there was no sign of loss of silencing efficiency when we introduced single nucleotide mismatch. So this is with the spike, and we did something similar with nucleocapsid transcript, where you see when you have single nucleotide mismatch, the activity was conserved, except for this first guide RNA, which is confirmed in the Western blot here. So the take-home message is that this Cas13 drug is likely to be resilient to the mutational escape that RNA viruses usually use to escape from host immunity or antiviral drugs. So the question was then, can this technology or this Cas13 tool silence replication competent SARS-CoV-2 or a real virus? And I'll just hand it to my colleague, Dr. Wei Zhao, who pioneered this silencing in infection assays.
Thanks, Mohamed, for your excellent talk. So I'll just continue. So, so what we did here at the Doherty Institute is that in the high containment laboratory, the PC3 lab, we infected those CRISPR-Cas13 expressing cells with real virus. So at first we used the Wuhan strain, the wild type, or the ancestral strain. So what we did is that we used two cell lines. First is a viral cell, which is a commonly used cell line for the coronavirus study, but it's a monkey. It's from monkey. So we also use another human lung cancer cell line, CRISPR-Cas13 cell. So after we infected these CRISPR-Cas13 expressing cells, then we collected supernatant at different time point post-infection and measured gene copy number of SARS-CoV-2 by PCR or the viral load in the supernatant by the infectivity assay. So as you can see here, first at different MOI, which means the multiplicity of infection, we saw a dramatic suppression of viral gene copy number in cells with CRISPR-Cas13 that targets the nucleocapsid protein of SARS-CoV-2. So at both 24-hour and 48-hour post-infection, and there were a significantly less or lower gene copy number of SARS-CoV-2 compared to cells expressing non-targeting CRISPR-RNA. And here with a lower MOI or low with or fewer initial viral inoculum, we also saw a very almost a complete suppression of viral replication at 48-hour post-infection. So if we just look at 24-hour post-infection here at both MOI, apart from the nucleocapsid protein CRISPR-RNA and other CRISPR-RNA that targets different sequence of the SARS-CoV-2 virus, like the spike sequence or the non-structural protein 7 or 8, they all showed very similar, but still like very effective inhibition of viral replication compared to cells with non-targeting CRISPR-RNA. Another assay we did is the infectivity assay. So as you can see, very similarly, cells expressing CRISPR-RNA that targets nucleocapsid protein, but not non-targeting CRISPR-RNA showed a significantly lower viral load at 24-hour or 48-hour post-infection. And here with at MOI 0.01, slightly lower viral input, we saw almost a 2 log less viral load in the supernatant in cells with CRISPR-RNA targeting SARS-CoV-2 nucleocapsid protein. When it came to Cas13 human cell line, again, we saw almost complete inhibition of viral replication at 48-hour post-infection in cells expressing a nucleocapsid CRISPR-RNA. At that time, the strain of concern or the variant of concern is the UK strain. So what we did is we just substitute the Wuhan strain with the alpha strain. So as you can see, so very similarly, cells expressing CRISPR-RNA that targets nucleocapsid protein could still effectively inhibit the replication of the alpha strain at 72-hour post-infection. That's confirmed by qPCR and viral infectivity assay, and showed that actually at 48-hour post-infection, viral replication had been suppressed, and at 72-hour post-infection, there was almost more than 1 log down of the viral load in supernatant in cells expressing CRISPR-RNA that targets the nucleocapsid protein of SARS-CoV-2. As Mohamed just explained that CRISPR-RNA and Cas13 CRISPR-RNA could tolerate one nucleotide mismatch. So what we did is that we next infected CRISPR expressing cells with both ancestral, the wild type, or the D614G mutant virus, and then measure viral replication by qPCR. So first, when we infected the cells with the ancestral or the Wuhan strain, those CRISPR-RNA, they still perfectly match the sequence of the wild type virus. So all the CRISPR-RNA transfected cells showed like before, dramatically suppression of the viral replication at 48-hour and 24-hour post-infection. Interestingly, when it came to the D614G mutant virus, now because those CRISPR RNA that targets spike, they possess a single nucleotide mismatch at the D614 position. So we saw a slightly less suppression, but still significantly lower of viral gene copy number in cells expressing these spike CRISPR RNA. But however, we could see that because the nucleocapsid CRISPR RNA is not affected by the mutation, it still perfectly suppresses the viral replication like before. All right, with that, I'd like to just summarize on behalf of Mohamed. So reprogrammed CRISPR-Cas13b effectors achieved more than 98% of silencing efficiency in a transfection model or a virus-free model. CRISPR RNA multiplexing suppressed viral replication by up to 90% in mammalian cells infected with real SARS-CoV-2 virus. Last but not least, single nucleotide mismatch, D614G, does not dramatically affect the capacity of a single CRISPR RNA to suppress SARS-CoV-2 replication. So just very briefly, the future direction of this project, we actually hope we could deliver CRISPR-Cas13 to primary cells and then animals and models, and then of course to human being. So what we could use is probably the lipid nanoparticle, as it's been shown to be very effective with CRISPR-Cas9 for treating rare disease. It's been published in the New England Journal of Medicine recently. So we eventually want to develop an aerosolized CRISPR-Cas13 lipid nanoparticle and turn it to an anti-COVID spray. Another take-home message here is that CRISPR-Cas13 is readily adaptable to other pathogenic viruses that may emerge in the future, especially RNA virus. So before I stop, I'd like to thank so this acknowledgement is from my view. So I'd like to thank, of course, first Mohamed, Fareh, and Joe Trapani for their just great idea and everything they developed in their lab, especially the CRISPR RNA and all the screening. I also want to thank my supervisor, Sharon Lewin, for her great introduction talk, and also all the lab members as well as our collaborators at the Doherty Institute. I also want to thank all the members from Vidro providing us these viruses, as well as our international collaborator in Hong Kong University. And also I'd like to thank Colin from Monash University. Thank you very much. I think that's it for three of us.
Thank you so much. Thank you for those excellent talks and really fascinating work. Congratulations on such very nice work. I've got a question, first of all, for Mohamed. Mohamed, would you like to comment on whether there and if there currently is a potential diagnostic application for this technology that you've spoken to us about? And also, maybe you could also tell us whether there's an optimal size or region that you've discovered for targeting this Cas13. So 2 questions there for you, Mohamed.
Yeah, thank you for the brilliant 2 questions. So as for diagnostics, there is a lot of work done in this space using either Cas13 or another CRISPR type called Cas12. That allowed scientists, mostly in MIT in Boston, to do very precise and rapid diagnostics of SARS-CoV-2, but also on other pathogenic viruses. So the second question is about targeting, which region we should target, how can we design guide RNA? and so forth. And I think this is one of a research area we are very active at. So we work with this Cas13 called Cas13b, and we been able to discover really key molecular mechanisms that allowed us to reprogram this protein efficiently. So, for example, what we found, we looked at secondary structure of the target, the folding of guide RNA, the hybridization energy between the guide RNA and the target. And the key finding we found is that the spacer nucleotide composition is extremely important for silencing. So there are some micromotifs within the guide RNA that tells you if this is going to be very effective guide RNA or very poor guide RNA. And these were possible because of bioinformatic analysis and single nucleotide screens we deployed.
Thank you for that. And I guess a question for Wei, could you tell us whether any CRISPR drugs have made it yet to clinical trials, either in cancer or in any other fields? I think you mentioned amyloid, but are you aware of any others where this has come into clinical practice? Or maybe that might be a question for Sharon as well. Wei, would you like to start?
Yeah, of course. So as I shown in my, I think, last slides, that the CRISPR-Cas9 drug for a rare disease, I can't remember off the top of my head. It's been, I think it's been tested and it's been definitely tested in a clinical trial, and it's been published in the journal. So and it's used this lipid nanoparticle to deliver. So I think it's really promising. And CRISPR-Cas9, they just they move a bit faster than CRISPR-Cas13 as it's been discovered for a few years. So and also there's definitely other CRISPR-Cas9 drugs on the way. So in the future, we will see hopefully more CRISPR-Cas9 and also CRISPR-Cas13.
Thanks. Sharon, did you have any additional comments? So one other question for you was about the monoclonal antibodies. Do you see them developing further to become more accessible or easier to administer? So I guess two questions there.
Yeah, thanks, Monica. So no, not much more to add. There's other gene editing tools. I don't think any have been approved. The big challenge, of course, is delivering CRISPR, which has been altered by the mRNA lipid nanoparticle technologies. That was the study that Wei presented. There has been some ex vivo CRISPR interventions done with HIV. It was published in a case called the New England Journal last year. And in fact, the FDA has just approved the first in vivo CRISPR HIV study. So CRISPR-Cas9 for HIV is relevant. HIV is an RNA virus, but it reverse transcribes to become DNA. So you can use CRISPR to remove integrated DNA. But I think the pace, the real area that's moving super fast here is in vivo delivery systems, and which will revolutionize, as opposed to ex vivo modification and infusion of modified cells. The other question was the antibodies. You know, antibody, we're very excited about CRISPR-Cas13 because, as Mohamed said, you just need the sequence of the virus. So many of you will know that the mRNA vaccines were also designed on January the 11th, the day after the coronavirus vaccine became public, because all you need for an mRNA vaccine is the sequence of the virus. So that has tremendous potential. But antibodies also came out very, very quickly. Like that first antibody from Lilly was licensed in June or July of last year. I think what we'll see is better iterations of being able to give it a huge advance if we could give it subcutaneous or intramuscular. And also there are now some in vivo gene delivery mechanisms for antibodies. You can actually use viral vectors or nanoparticles, but there's quite a lot of advance with viral vectors, adeno-associated viruses, to deliver the gene for the antibody rather than giving the infusion, which is quite clunky and obviously intravenous and expensive. But I think the antibodies for COVID, ideally, if you want, I was trying to emphasize the need for an easy, low cost, low toxicity intervention, because remember, you have to treat lots of people with early symptoms to prevent a few of them progressing. And antibodies, the number needed to treat is high. So at the moment, it's not so cost effective. And just but for people to know that we are giving antibodies in Victoria to people with less than 5-day symptoms, older and with comorbidities. But I think we'll see a lot of improvements in antibody technologies too in the coming months.
Sharon, so a question for Mohamed is about whether you've thought about using AI to assist in finding the best RNA guide, and also in terms of when you're developing this drug, are you looking at a multi-target? So looking at both the spike and the nucleocapsid, or multiple targets in the one drug?
Yeah, that's a really great question, and we haven't had the time to go through the data. We to start with the second question of multiplexing that drug to target several genes. We have done this in the paper, and so the idea behind is by targeting several genomic regions. So even if the virus mutate and is to escape from, one location, the fact that you are targeting several locations makes it unlikely for the virus to escape from this therapy. And we have done like pool of guide RNA targeting the spike, nucleocapsid, and RNA-dependent RNA polymerase simultaneously, and we see great effect of silencing with this pooled guide RNA. It's really nice idea that would work in virology, but also in the cancer space, as we know that cancer cells can also escape from single drugs. So the second question is related to artificial intelligence. And this is something we are working in. So now with CRISPR screen technology, we can generate thousands and thousands of data sets that the single mind cannot process in efficient way, especially when you are looking at different parameters like folding and hybridization and so on. So for now, we are still doing this at old fashion. But we are looking to the use of artificial intelligence and to basically see the unseen in our data, and we will come in collaboration with people that are expert in this field.
One last question is probably a question for Sharon. If this drug becomes licensed and there's a slight mutation in the virus that requires a adaptation of the Cas enzyme, do you foresee that it would have to go through the whole clinical trial process again to market, to bring into practice a slight variation of the drug?
I kind of am watching this space with great interest in the mRNA vaccine world. So I think it's kind of analogous here. So with as many of you will be reading, Moderna are making mRNA boosters using a variant sequence, using the sequence for the beta strain, South Africa, as opposed to the Wuhan strain. And so they, of course, have to show safety. So there's still phase 1, phase 2, and immunogenicity of their new target mRNA, but they don't have to go through the large clinical phase trial to show efficacy. So I think it would be, they will have to show efficacy as a booster, of course, with these new variant vaccines. So I think it's a little bit analogous. And I think maybe Mohamed can, I don't think you can get resistance to Cas13b. You can get resistance to the guide for it to bind, but I don't think you can get resistance to the actual enzyme. And our strategy will be to target highly conserved areas, because there are parts of the virus that can't change, otherwise it can't replicate, and also to multiplex, so that resistance should be rare. But it's a good question. I probably think we would use the same approach as what's happening in the mRNA vaccine world now.
Yeah, exactly. So I think by targeting a highly conserved region and having pool of guide RNA instead of single guide RNA, the likelihood of the virus escaping is null. And this is similar to the tree therapy in HIV space, for example. It's impossible for the virus to accumulate the exact same mutations to escape from this pressure you apply on the virus.
Well, thank you very much. I'd like again to thank all the speakers for fascinating presentations, and I really look forward to hearing about the development of this drug. So that brings us to the end of today's webinar, and a reminder that today's webinar will be recorded and will be available on the VCCC Alliance Open Learning page once the data has been published. So I would just like to thank again all of our excellent presenters today, and thank you to you, the audience, for tuning in. Thank you, and see you at the next one.
Thank you.
Thanks, everyone.
Thank you.